Today you will learn the basic physiopathology of human disease, X-linked nephrogenic diabetes insipidus. This is a genetic disease for which we still don't have proper treatment. That's why at the end of the presentation I will introduce you to a relatively new technology that I think in the near future will help scientists to find new pharmacological treatments for this disease. Now, X-linked nephrogenic diabetes insipidus is a disease of the kidneys, and it particularly affects the kidney's ability to retain water from the urine. As you all know, we are about 70% water in our bodies, so it is extremely important for us to have a proper system of controlling the amount of water that we have in our cells. The body does that by by the hormone-based pressing, which is excreted by, this doesn't work, by the pituitary gland, which is at the base of the brain, right here. Whenever the amount of water that we have in our bodies is reduced for any reason, the pituitary gland secretes the hormones-based pressing, or also ADH. This hormone acts in the kidneys, which will retain more water from the urine. Let's go to the kidneys to see more detail what happens. This is a drawing of a nephron, which is the functional unit of the kidney. The blood is filtered in the glomerulus, and then the filtrate travels along these tubes where the urine is formed. At the end of these tubes, you have the collecting tubule, which is where the final processing of the urine takes place. In this area of the kidney, we have the cells that respond to the hormone-based pressing. This is a drawing of the principal cell. You see it has two sides, an apical side and a basal side. The apical side is the internal side of the tube, which is in contact with the urine. The basal side is the external side of the tube, which is in contact with the extracellular fluid or with the plasma of the blood. But when the hormone reaches these cells, it binds to its receptor, the big 2 receptor, and when that happens, some mechanisms happen that increases the intracellular concentration of a chemical, cyclic AMP. This chemical is a secondary messenger which signals the other side, the internal side of the cell, to move these vesicles to travel to the plasmatic membrane and insert a water channel in the membrane. The name is aquaporin 2 channel. These water channels will cause water to move from the inside of the tube, from the apical side to the basal side. In that way, the amount of water in the urine is reduced, and the body is able to preserve water. Now, XNDI patients, or X-linked nephrogenic diabetes insipidus patients, lack the big 2 receptor, so they are unable to respond to the hormone, and that's why they lose higher amounts of water in the urine and are in constant danger of dehydration. Now think for a minute, if you want to discover new pharmacological treatments for this disease, which kind of effect would you like your drug to have? Obviously, one of the effects that you will be looking in your chemical is a chemical that could exert the same mechanism, the same response in your cells. In this case, one possible mechanism that you will like being exerted by your chemical is an increase in cyclic AMP. And this hypothesis has already been tested and it works. Now, we don't have enough chemicals to test in either cell models or animal models before we can find actually one that could work with humans. And that's why I believe that a technology that allows us to discover, to identify which chemicals from all the chemicals we know could cause this effect in the cells would be very helpful to find new treatments for this disease. And I'm going to introduce you now to these technologies, called high throughput screening. What you see in your slide, is a plate, a microplate, which is what we use to test several chemicals at the same time in your cell model or your biological model. So this is a microplate. It has, for example, this one has 1,536 little holes or wells. And in each one of them, you can test one different chemical. And you have other different microplates as well. You have other different microplates as well. And so this technology has been possible in the last, let's say, 10 years because of advancement in technologies that allows us to manipulate fluids in high quantities and with precision. So that's why in these facilities they use robots and equipment you see here, that the microplate is under the device that handles all the plate at the same time. And this technology is really great in terms of testing thousands and thousands of compounds in a short time. And this will be the outcome that you would see in your computer. You see that in each well you have a different color, meaning a different concentration of cyclase P, for example, if this is what we're testing. So each drug we have a different amount. Before we finish, I'm going to show you a few seconds of a video so you can see how in a regular facility that I would say here.
Wait, pause the video. Pause the video. All right, now talk. I'm going to show you a video.
I'm going to show you a video of the functional of a facility in which they do high throughput screening, so you can see the robots working and handling the different microplates.
There you go.
Just about 15 seconds. That allows scientists to let the facility work by itself and handle all the load of testing automatically. So these robots put the plates in one place and move them to the place and incubate the plates with the different compounds by themselves.
And what are you doing while this is happening?
And you're just controlling the situation. You're just checking that no errors are happening. So as a conclusion, this is a conclusion I didn't modify at all. XNDI, or X-linked nephrogenic diabetes insipidus, is a human genetic disease of the kidney caused by mutations of the gene of the vasopressin 2 receptor for the water-conserving hormone vasopressin. Chemicals or drugs that can increase the biochemical second messenger cyclase AMP in the kidney polyphenolic cells, which you saw in the drawing, mimic the effect of vasopressin on these cells, and consequently could potentially be used as therapeutic agents to treat XNDI. Findings of chemicals compounds could be significantly stimulated by the use of high throughput technology. Thank you very much.